welcome to today's lesson. This is Mr. Geno, and I want to present for you the DNA and protein synthesis. This is the topic that I want us to talk about today, and we want to base our argument on what we learned in the past. So we have been talking about the genetic coding, and today I want us to see how the DNA is involved in the protein synthesis. We know that our body cells is mainly made up of protein, that is a quite percentage of it. The enzymes that we need are made up of protein, and this protein's molecule must be synthesized within the cell. Now we know that in the eukaryotes, that is the organisms that have got cells which has got the nucleus that is membrane bound, we know that the DNA that code for the individual protein is in the nucleus of the cell. So that is the first thing that we should be capturing, that the DNA and that code for a specific protein is found within the nucleus of the cell. This is one thing that we should understand, that the DNA that has got the information about a specific or individual protein is actually found within the nucleus of every cell that normally will be giving us the information of how this particular thing can be produced. But we know that the synthesis of the protein, synthesis of protein is done by ribosomes. Which is actually found within the cytoplasm. So ribosomes found within the cytoplasm of the cell. So this one is now taking us to our main key thing that we should be basing our argument from, that this is what has got the information about the protein to be synthesized. Its location is the nucleus, and then we have got the area where this particular protein synthesis will actually be taking place. That is the ribosome, which is found within the cytoplasm. So the next question that we will be asking ourselves, how are these particular things linked? So we really know that in terms of this particular analysis, we have not detected any DNA within the cytoplasm. For the eukaryotic organism, the DNA is not found within the cytoplasm. Therefore, there will be no direct synthesis of protein when this DNA is going to be in the cytoplasm. That kind of direct synthesis of protein is actually missing. So we know that the synthesis of the protein is always carried out by a transfer of information from the DNA, which is found within the nucleus, to the ribosomes. So in between here, we need the materials that will be carrying the information that will help us to actually synthesize the protein itself. And this particular material that is going to help us in relaying this information is called the RNA. The RNA. This is what is going to help us in transferring this particular information from the cells, from the nucleus, so that they can be taken to the DNA where they should be found. Now the first thing that we should remember about the RNA is, how do we compare the RNA and the DNA? So the first thing that we learned is that this particular molecule, the RNA here, is more of the same as the DNA, because it has got the phosphate molecules, it has got the sugars, and then it also has got the nitrogenous bases. But the only thing that is now becoming to be different here that we had studied early enough is that RNA, normally in the nitrogenous bases, it has uracil. If you can remember, it has got uracil instead of thiamine. So whenever there is a thiamine in the DNA, in the RNA, it is replaced by uracil. The rest of the bases are the same. So that is one thing that is bringing in the difference between the DNA and the RNA. Now RNA has got the ribose sugar Remember, the DNA has got the pentose sugar. So this one has got, and the deoxyribose sugar. This one has got, you can say, it has the ribose sugar. These are differences that we can locate between the RNA and the DNA. Then finally, this one here was the DNA was double helix. This one is single-stranded molecule. Single-strand molecule. So these are some of the key things that we have to highlight that brings in the difference between the RNA and the DNA. So we have to realize that for this particular protein to be synthesized, number one thing is that the information must be collected from the DNA within the nucleus. So that one will be taking us to what we are calling types of RNA. We have got different types of RNA. Types of RNA, and this is what will be helping us to understand the protein synthesis. So I want us to go with it slowly so that we know the types of RNA. So we have seen that there is one thing that must be there about this particular transfer of the information, or the synthesis of the protein. RNA is going to enable the information to move from the nucleus to the ribosome, where the information will be, where the protein will be synthesized. So there's specific 3 processes that are involved during this particular process of protein synthesis. The first process, that the RNA will be carrying in the instruction of the polypeptide from the DNA in the nucleus, and then it carries it to the ribosome. So that is the first thing, making sure that the instructions that is found in the polypeptide from the DNA that is found in the nucleus is carried within the nucleus to the ribosomes. So that is the first role that it will be doing. Number 2, it is going to pick up the specific amino acid from the protoplasm of the cell, and then it carries them to the surface of the ribosomes, where the protein synthesis will be taking place. So that particular specific amino acids that is responsible for binding together to give us a long chain of the protein that we need is carried within the protoplasm, and then it is taken to the surface of the ribosomes, whereby they are going to be linked, and then they give us the protein that we are looking for. Then the last thing is that it makes up the bulk of the ribosomes themselves, so that the ribosomes can bind all these particular amino acids, so that we give us the specific protein that the body needs. So that is how this protein synthesis, those are the processes that are involved in the protein synthesis. So for the first process here, we must understand the first type of RNA that is going to help us in doing this. So we are calling it messenger RNA. We basically write it as a messenger RNA, a small m down here. This is what is going to help us in the understanding the first stage of the protein synthesis. Now, this particular messenger RNA is formed in the nucleus, and then we know that the piece of the RNA is usually instructed from the polypeptide. So we know that we have got the DNA in the nucleus, and it is a double helix. So the first thing that normally happens that we had agreed on in that particular DNA, it will unwind, so that when it unwinds or it unzips itself, it is going to give us the 2 strands. We have got what's called the sense strand and the antisense strand. The antisense strand is what is called the template strand. So the template strand is where, that is in the DNA, that is where the messenger RNA will be aligning itself, so that it can receive, it can pick up the codes that are responsible for certain protein synthesis. So it is going to align itself in that particular template strand, and then after aligning itself there, and due to the base sequence that we have between the DNA, the RNA will be able to pick up that particular information. And during this particular time, the picking up of the information will normally start from the starting code, which we have talked about it in the previous lesson. That is the methionine. That is the first thing that is going to start from. So it picks up those particular information, and then during this particular picking up of the information, there is an enzyme that helps in this particular molecule to align itself within this particular, the template strand of the DNA. That is what we are calling the RNA polymerase. So it is going to join up the enzyme polymerase help us to join the chains of the RNA nucleotides together, so that at least they can line itself in the template strand and then collect the information from the nucleus. The moment this one is done, this particular messenger RNA will always separate from the DNA template, and then it allows the DNA chains to double helix to rejoin together using the hydrogen bonds. Remember, it had opened up. So once it has given the information, and then this particular RNA will come out with the information so that it can relate to the next cells, to the next stage. So as it is doing that, the DNA will not remain open like it is. So it is going to unzip. It's going to zip up again, and it is going to zip up by rejoining itself through the hydrogen bonds that were opened up during the time when it was being separated, the arms themselves. So that is the first thing that normally happen. So this particular instruction that have been taken by the RNA, they are in the same way they were in the DNA base. So there is those particular, the triplet codes that the messenger RNA is having with it, and as it is moving with it outside the cytoplasm. So all this scenario should look like this. Let us summarize it in the diagram so that we can understand what it looks like. Now this is the diagram of how the messenger RNA is picking the information from the nucleus of the cell and then transmitting to the cytoplasm, whereby it is going to be picked by the next ribosome, the next RNA. Now this is the entire double helix strand of the DNA, and this is what we are referring to the sense strand, where we have the codes for the protein to be synthesized. Then this is the antisense, the template strand of the DNA. So you can see that the first thing that has happened is that the DNA has unzipped. It is opened. This one enables the RNA, and in this case messenger RNA, to align itself on the template strand, like you can see how the bases are being aligned. Now the first thing that you can see from the beginning, we can see that this is the adenine A in the DNA strand, which will be matching with the other DNA strands at a T. But for the RNA, it is going to match with a U. Then a C is matching with a G in the RNA, and also on the other side it's C, how it's matching. So this is how there is that alignment of the messenger RNA within the template strand. And I say that this one was done by the RNA polymerase that is going to link these particular molecules together so that they can the RNA strand can align itself all together until then, so that it can pick up that information. So that is the first thing that was happening there. Now after that, this particular thing will go. It will come out of the cell like it is being illustrated here. This was the RNA in the nucleus. So it has picked up the information, and then the nuclear envelope open up through the nuclear pores so that the RNA can move out of it. So in the cytoplasm, there are a lot of amino acids here that we have here that we are going to see how the next RNA will be working on that. Now after that, we'll see that this thing will be zipping up again so that it can wait for the next process to continue. So that is how the messenger RNA works, so that it can pick up that particular the information that is going to be relayed on the protein synthesis. That is now what was happening. Now from there we have got what is called the transfer RNA. So the transfer RNA is basically found within the cytoplasm of the cell. So that is our next the next RNA. We call it a transfer RNA. Now that is what we have. So what is coming from the cytoplasm here, the messenger RNA is coming up with the code that is supposed to be used to make the protein. Now when we reach in the in the cytoplasm here, I have said that we have got amino acids within that particular area, and then we also have got the transfer RNA, which is actually having the anticodes RNA It is the anticodes of what was picked by the messenger RNA within the nucleus. So what it is going to do is that at this particular point here, this particular transfer RNA is able to collect the amino acids that were found within the cytoplasm of the cell, so that they can be bonded together in a sequence that will be matching the code that this particular messenger RNA has come with, so that they form the anticode of what was carried within the messenger RNA. So this one is able to bind, so that we have got a binding site. Like we can see, this is the tRNA that I've drawn for you. There is the binding site of the amino acids that are being collected within the cytoplasm, so that they can form the anticodes of this particular information that has been collected from the nucleus of this particular cell. Now, when these particular two things are, the amino acids are collected here within the cytoplasm, and then they are collected together, and then they are binding on the surface here, so that they can be forming the anticodes of the messenger RNA that has come up with. This activity is going to be done, basically, the pieces that are going to be brought together will be helping us to determine the exact protein that will be synthesized, so the exact amino acid that we really need. Now, all this binding is going to be done in the surface of the RNA, whereby the actual protein synthesis is going to be synthesized. Now, once that one has been done, we have seen the sequence, how it is coming in, that the base sequence here are aligning themselves, and then they are brought together to form amino acids that have been collected from the cytoplasm. That one is done at the surface of the ribosomes. So what is done, what we are left with is to actually translate that information that has been brought from the nucleus, and the anticode has been collected together. So we can translate and actually instruct the ribosome on which specific protein do we need at this time, which enzyme do we want to produce. We already have collected the information, the required amino acid. So the next thing is, we want to see which is the protein that we are going to synthesize, which is actually done within the ribosome itself. So there is that actual translation that is done in the ribosome. So this one here is done by the third RNA, what we are calling the ribosomal RNA. They are basically found within the ribosomes themselves. So they are able to bring together the large molecule and the small molecules of the amino acid that were collected by the transfer RNA that is now found within the surface there, and then they translate them, so that we really know which particular amino acids do we want to get, by binding those particular small amino acids to form a very large chain. And this one is done from the beginning of the codes, what we were calling the amino acid that we talked about, until where we have got the stopping code, so that we already have got a large chain of the protein that we need. So this process is continuous, so that we get the exact protein that our body is really supposed to be producing or having. So that is the process that I really want us to really understand, and then we know that is what we are calling the protein synthesis itself. So the protein synthesis is actually a process that starts within the nucleus itself of the cell, whereby the DNA is supposed to be exposed, so that we have got the RNA, the messenger RNA, collecting the information or the codes that is needed for the synthesis of the protein, and that one is found within the template code. So when the messenger RNA come up with it, there is it brings those particular code out of the nucleus, and then I will say that within the nucleus we get the transfer RNA, whose work is to, first of all, collect the amino acids that are found within the cytoplasm, and then it has got the anticodes that is going to help us in translating the information that is coming from the nucleus. So it is going to bind with this in the surface of the cell, and then during the ribosomes, whereby there is actual translation, and this one is done by collecting all the amino acids that have come with it from the cytoplasm to form a long chain, so that they give us the specific protein that the body needs to produce. So that is what we normally have, and then these ones will be resulting into the protein synthesis, which is done by actually ensuring that the hydrogen bonds that were broken here are brought together to help us in forming a very long chain of the nitrogenous base that is going to give us a specific protein that our body needs. So that is how the protein is synthesized. So please, I want us to really understand the steps. What is the activity or the event that takes place in every step, and what is the resultant product that is actually coming up with. So that is how the protein is synthesized in the DNA. That is the role of the DNA in the protein synthesis. So please, viewers, I want us to be able to link the two. If the diagrams are brought to us, we have to know that in each stage what happens. Like, remember the first stage in the diagram, what we were talking about was the transcription of the codes. That is, the DNA molecules was unzipping, and that is was to expose for us the two strands, the sense strand and then the antisense strand. And that one was able to make the messenger RNA to align itself in the antisense strand so that it can pick up the code. That was the first event, what we are referring to as the transcription. Now from that, we are supposed to make sure that the nuclear pore will open up to allow the messenger RNA to actually come out with this particular information into the cytoplasm. And then into the cytoplasm, we have got amino acids that is being activated, is being carried together by the transfer RNA. So that is the first event that is coming up. This particular amino acid is the one that's going to help us to come up with the specific protein that we need. So we have to collect them. And that was the first thing that the transfer RNA was doing to us by collecting this particular amino acid. That is the event that was actually taking place. Now, the moment they have been brought together, they were being linked together through the hydrogen bonds that we have got the long chain. Actually, that is now the translation, which is done by this particular the tRNA that is found within the cytoplasm. It brings them together. After bringing them together, it enables the ribosomal RNA that can be bringing these things, the amino acids together, so that we have got a specific long chain that is giving us a specific protein that we want. So that is now the transcription, what should be happening, and then we have got, at the end of the day, a known protein being synthesized. So that is how the protein synthesis is taking place. Now, welcome to this session. I want us to see this particular idea of the gene mutation. Now, we have known what a gene is and how it is used to transfer information from one cell to another, or from an organism to the next organism. That is one thing that we have seen. So when we talk about a mutation, we can describe it as a change within the base sequence of the code that gives us the information. So it is something that is abrupt, that occurs in the bases. Now when it occurs in the bases, it is these bases that give us a sequence that will be coding for a specific information. So the moment there is a change in any of those, that particular final thing that's going to come out of what was intended will be a changed information. And that is now what is called the mutation. So this one normally occurs when cells are dividing. Either the normal body cells are dividing, a portion of the nitrogenous base in the gene can be removed, or something can happen to it that can bring in a change. Or it can also occur in the gametes when there is meiosis. So when it occurs in the cell body, when the mitosis is taking place, it means that a portion of the organism will be affected. And when it occurs in the gamete, it means that it might even be severe, because in the gamete, it means that we are affecting the generation to come, not the current organism only, but we are affecting the generation organism to occur. So that is how the mutation itself looks like. Now we know that cells themselves also have got a mechanisms of helping, or what's called the anti-mutation that occur in the body. We have got a way of making sure that we can correct this. That is when the change is very much negligible. But we can have the case whereby the mutation can be severe. So we have got types of mutation. Mutation can occur at a point or in the gene, what we are calling the gene mutation. So that is in a specific non-nitrogenous base can be affected. Therefore, the code for certain information will be altered. Or it can occur in the chromosome. So we can see those particular type of mutation, how it occurs. So I want us to have a look at when this particular mutation occur in the point, that is at the gene. What are the effect, or what are some of the things that can be seen happening in this particular manner? Now, this particular gene mutation, we have them occurring in different ways. So I want us to have a look at the different types of the mutation that can occur in the gene. So I have got the base sequence that we are having here that is coding for a specific information, or for a specific amino acid to be synthesized. So I have got this is my base sequence, a TAA, that is giving us a code for an information, and then we have got the TGC, that is what originally we have. So a change can occur in a specific nitrogenous base. Like I want to look at the first change that we are having here is called insertion. Now insertion, just from the word English, means to insert, to bring in between. So this is a case whereby in terms of the gene being transcription is happening here in the genes, there is a specific nitrogenous base that is inserted in between the original bases. So that one is going to give us a new information. So if you look at it at the end of the day, the mutated gene now will be looking at this. So we see that there is a new gene here that has been inserted in between. This is what we originally have, and then there's a nitrogenous base that had been inserted there. So that one means that we now have got a new code with a new information. So that is one of the things that can happen there. Now we can also have got what is called substitution. Now substitution means that you are taking what is you are having in there and you remove it entirely and then bring in a new thing again. So you are replacing. So if I've got this particular base sequence here, I can have got the substitution on this particular gene. So the gene A is removed, and then in its place we have got gene G coming in. That information has been changed again. So that is some of the things that can happen to our body. We have got deletion. This is from the word delete. You entirely remove a section and you don't replace it. So again, that one is going to give us a new sequence that is going to code for a new information. Like in this case, we decided to have the deletion on the A. So it is deleted. So this is now what we have in the information. So that is how the gene mutation can occur in our body. So that is one type of mutation. Then we also have got a type of mutation called chromosomal mutation. And we have looked at the gene mutation that it is at a point whereby the specific nitrogenous base has been interfered with, or it has been affected. Now in a chromosomal mutation, it involves the change in the portion of the old genes within a chromosome. So we can affect the old gene that is found within a chromosome. Now this particular change here can also still have got some verse effect. So we can think of like for example, if we have got a word in a sentence and then we change a word in a sentence, you will realize that that particular change can make the word to have got meaning, or can still retain meaning, or the meaning can be distorted completely. So that is how it can happen in a chromosomal mutation. So chromosomal mutation is a change that is found within the chromosomes, that is the genes, the way they are arranged in a chromosome, and then when it is going to be transferred to the next organism or the next cell, you can realize that the change can be noticed or the change might not be noticed at all. So that one depends on how the change was, but the fact is there is an entire change. So we can have the change in the whole chromosome. Like for example, we have got a case whereby a certain number of chromosomes is affected in an organism, like the case for the people who suffer from what's called the Down syndrome. In that particular case, the cell has got a chromosome of a specific number, like in this case, number 21, is actually mutated in that this chromosome can be repeated. So you'll find that in the organism, the whole organism here, the individual which have got this particular syndrome here will be having a copy of this chromosomes. 3 of them, instead of having the normal 2 when they are paired. So when this particular is interfered, the chromosome is interfered with, this organism will be experiencing this particular problem that is found in them. So that is the thing. So this particular mutation can affect the physical appearance of organism, what we are calling the phenotype. So when it brings in this, it brings in what we are calling variation. So variation is actually the observable differences in an organism. Those particular differences that you exhibit from the normal thing that you have, and then maybe the next generation, what you're expecting from them, there is a slight change that we can notice. Now that is what's called variation. Now this particular variation can be expressed phenotypically. In other words, it's something that can be seen in the outward appearance of an organism, and that one here is also having an effect of an organism. So that is one thing that we normally have seen. So we have got the conditions that can bring in this particular change, and these changes, some of them can be advantage to some organism. If any case they enable the organism to adapt well in the environment, so they can make to increase the reproduction of an organism. Like for example, if any case the change is bringing you to adapt well in the environment, therefore the species that come out of that particular new character can survive very well. And this one is going to give that organism an advantage of reproduction by making sure that each species are more retained in the environment than the ones that are lacking that particular feature. So that variation here can be an advantage, or it can be a disadvantage if any case it doesn't bring in the survival aspect part of it. So that is one thing that we are experiencing with this particular chromosomal mutation. So it can be causing a vast effect, or it can be having a minor effect that we can have in the environment. Now this particular mutation can be random. We have got the random mutation in the genetic materials of organisms that can be within the gamete, or it can be within the body cells of an organism. But in some cases here, if any case we have got this particular mutation during the cell division that we have been talking about, we have got various problems that can occur. We are talking about the Down syndrome that can occur when a specific chromosome is affected. The organisms will be having a problem. We can also have conditions that come as a result of mutation, like what's called cystic fibrosis. This is a condition in which the protein does not function properly. So this condition is really severe if any case the gene that is responsible for it is mutated. So it can bring in that condition in the environment. And then we also know that in some particular instances, the organisms may escape mutation. This is because we have got what is called the non-coding gene. So when we talk about if the mutation occur in the part of the non-coding gene, that one, the genes that which does not affect the genetic codes is red. So if we have got the non-coding genes here, they don't bring us the code that will be responsible for specific protein synthesis. So if any case they are mutated, the effect might not be severe. So that is one thing that can help us if any case the gene is occurring in an area whereby the gene cannot read it, so that it cannot be interpreted, and then it gives us the specific protein at that particular point. So that is now a point whereby we can escape that particular type of the severe end. Or we can also have what the code is degenerated. In other words, it is small, and then the code can now not alter the amino acid code itself. It cannot change the amino acid that's supposed to be synthesized by that particular point. So it can say that it can just be degenerated, it can just disappear like that. So that is one way by which the gene, the chromosomal mutation can be not severe in some instance, or it can be severe. So it's based on what has happened. What is the specific gene that has been interfered with? Like for example, we can look at the instance of the sickle cell disease. What happened when the coding goes wrong? So I want us to look at the sickle cell anemic condition. Remember, a sickle cell anemic condition is a condition whereby we have got the normal hemoglobin of an individual. There is a normal hemoglobin that carries enough oxygen from one point to another. If any case an individual has got sickle cell anemia, it means that this particular hemoglobin, the gene that is responsible for the synthesis, is actually altered, and therefore the gene, the hemoglobin that we have will stiffen, and it forms the rod that change into sickle shape. Now, this particular sickle shape here that we have here, the effect that we'll be having is that the individual will be having the hemoglobin that is unable to carry enough oxygen, and even the enough blood that will be moving into these particular tissues that will be helping the respiration is not going to be there. So this individual will be suffering from the inadequate oxygen, and therefore it brings in some painful events to this particular person. So it is done when we have got a certain protein is replaced, a certain gene is replaced. Like we have got, we have talked about the collection of amino acids that help us in the protein synthesis. So if any case we have got it occurring in this particular pattern, like let us look at the sequence of the normal amino acid. So this is an example of a case whereby we have got the chromosomal mutation, and we can see it in the condition that we are calling the sickle cell anemia. And we have agreed that in the process of the protein synthesis, now the protein that will be helping us to give us the normal hemoglobin in a person, if any case we collect all the amino acids, this is what we should be having. We collect this is the code for the amino acids. So if we collect all the possible amino acids, we'll be having the normal or healthy hemoglobin in this individual. But let us look at what has happened in this particular individual. We have got, if we compare all of these amino acids that we have, they are all okay, except for when we reach here. If we compare the amino acids that we have at the beginning, we have got our A here has been replaced by a T. That is a mutation. And therefore, the code here that we are having is a new code which gives us a new amino acids, not the one that we were intending to have, the glutamine. So we have got the valine. So in this particular case, this particular individual will be having a problem with his hemoglobin. It's not going to be the normal shape hemoglobin. This is a hemoglobin which is going to be very rigid in shape, and is going to be small. Therefore, it cannot carry enough oxygen that this individual should be having, that is being transmitted to the cells that will be helping the process of respiration. So this individual will be suffering from that particular condition that is called sickle cell anemia. So these are some of the things that we normally experience. So we can have this particular mutation being very severe. This is how we can have got the cells that can be leading to cancer, when we have got a mutation that occur in those particular genes that are responsible for them. So what causes this particular mutation? Mutation is caused when we are exposed to certain conditions that can bring in a change. That is what has got the ability to penetrate into the cell. Like we have got the mutagens, like the X-rays. We have got things like certain chemicals can bring in a change, and then we have got the radioactive rays. Those ones can bring in a change in the individual's gene. So it is advisable that we avoid exposing ourselves to this particular mutagenic condition. Mutagens are those particular conditions that can cause mutation, and therefore it is good for us to avoid them in the environment. So this is an example of a mutation, and that is how the instances of the mutation can occur in the gene. Otherwise, thank you very much for this particular lesson, and next time we meet, we'll be looking at the patterns of inheritance of a gene. So whether we are going to inherit the mutated gene or the normal gene, we are going to see how that pattern of inheritance occurs.